Esteemed Crafoord laureates, the Crafoord family and ladies and gentlemen, today you will hear the prize lectures for the 2025 Crafoord Prize in Polyarthritis. And there are two brilliant lectures that waiting for you. But before the lectures and before the presentation of the laureates, I will just briefly tell you about the Crafoord Foundation. It was established in 1980 by Holger and Anna-Greta Crafoord. And the Crafoord prizes are jointly given by the Royal Swedish Academy of Sciences and the Crafoord Foundation. So the fortune that is the ground for this was established by Holger Crafoord. He was a single child of a single mother, and she managed to get him a very good education. He graduated first in his class from the prestigious Stockholm School of Economics, and was quickly headhunted by Rausing to a leading Swedish packaging company, where he quickly became the CEO at a very young age. He then was crucial in founding Tetra Pak, that you probably all have heard about. And most of us use it on a daily basis. But after some 10 years, Rausing and Holger Crafoord went different ways. And he had quite a lot of money, didn't know what to do with it. And then by serendipity, he met this guy, Nils Alvall, at the time consultant in nephrology and later became professor of nephrology here in Lund. And he had an idea how to make a dialysis machine that would actually work. There had been dialysis machines earlier, but none of them worked properly. So Holger Crafoord put all his fortune into this company, Gamla Brogatans Sjukvårdsaktiebolag, or the healthcare company of the old Bridge Street, and which became Gambro and the world leading in dialysis technology. And when he sold this, Holger and Anna-Greta Crafoord established these prizes, who are given on a 4-year cycle. So 1 year mathematics and astronomy, then geosciences, biosciences, and polyarthritis. 4 of these 5 prizes were intended to complement the Nobel Prizes in areas where no Nobel Prizes are given, with one exception, and that is polyarthritis. And the reason for that is that Holger Crafoord himself suffered from a severe rheumatoid arthritis. So the previous prizes have been given for autoinflammatory diseases, for T regulatory cells, and for example, for the first biopharmaceutical TNF alpha blocker that really revolutionized treatment of rheumatoid arthritis and many other inflammatory diseases. So this year we have 2 laureates that's made enormous advancement in our understanding of B-cell tolerance. And Professor Pan Hammarström, member of the Royal Swedish Academy of Sciences, will present Chris Goodnow, the first laureate. Please.
Thank you, Olle. Dear Crafoord Laureate, Secretary of Royal Swedish Academy of Sciences, CEO of the Crafoord Foundation, and ladies and gentlemen. My name is Kämpe Hammarström. I'm professor in clinical immunology at the Karolinska Institute and also a member of the Crafoord Prize Committee. It's my great pleasure to introduce one of this year's laureates, Professor Goodnow. Together with Professor David Namasi, Professor Goodnow has been awarded the 2025 Crafoord Prize in Polyarthritis for the discovery of fundamental mechanism of B-cell tolerance. Christopher Goodnow was born in Hong Kong and grew up in Washington, D.C., US, before moving to Sydney, Australia as a teenager. He trained in veterinary medicine and surgery, as well as immunology at University of Sydney and DNA technology at Stanford University. After receiving a PhD degree at University of Sydney, he joined the faculty of Stanford University Medical School and Harvard Hughes Medical Institute in 1990. In 1997, he returned to Australia as a professor and founding director of the Medical Genome Center at Australian National University. In 2015, he joined Garvan Institute of Medical Research and became the director in 2018. He currently holds the Bill and Patricia Retcher Foundation Chair at the Garvan Institute and is also professor at the Cellular Genomic Future Institute, University of New South Wales, Sydney. When Professor Goodnow was a young student, he became interested in immune tolerance, that is, how our immune system learns to recognize and tolerate autoantigens. Specifically for B cells, it had been proposed that the self-reacting B cells are either deleted or rendered tolerant. However, due to the rarity of such specific self-reactive B cells, studying their fate had been a major challenge. Inspired from the T-cell receptor transgenic mouse work in Professor Mark Davis' lab, Professor Goodnow had a bright idea to generate a double transgenic mouse model to track the fate of self-reactive B cells. This was accomplished by mating the two types of transgenic mouse. The first carried the normal foreign antigen, the foreign antigen, hence egg lysosome, that was expressed in a way that considered as self-antigens. The second mouse model carried rearranged immunoglobulin heavy light chain genes encoding a high affinity anti-lysosome antibody. So using this double transgenic model, Professor Goodnow demonstrated that the majority of self-reactive B cells actually did not undergo clonal deletion, but instead became functionally silent, and they could no longer secrete anti-lysosome antibodies. These findings indicate that self-tolerance could be achieved through mechanisms other than clonal deletion. These transgenic mouse models laid foundation for a very successful PhD thesis, numerous influential papers from his lab and other labs around the world, and major insights into B-cell tolerance. Importantly, this work also provided a conceptual framework for understanding how failures in B-cell checkpoints can lead to autoimmune diseases. I would just like to highlight one important paper published in 1991, where Susanne Hartley is the first author. I hope she's among the audience here today. In this seminal work, she elegantly demonstrated that both Chris and David were correct. So the member-bound self-antigen led to clonal deletion, were the secreted form of self-antigen-induced energy. So they were both correct. So at that point, it seems that Suzanne probably should have shared the prize too. Finally, probably less well known, that Professor Goodnow is not only a passionate immunologist, but also a professional surfer. So his passion and love for surfing has led the discovery for one of the best surf breaks in the world, Macaronis, Indonesia. I think he probably will talk about that. So without further ado, I would like to invite Professor Christopher Goodnow to the stage to deliver the 2025 Crafoord Prize in Polyarthritis.
That's lovely. Thank you, Qiang. That was a really very thoughtful introduction, and I'm glad you covered the surfing, but most importantly, Suzanne Hartley's contribution. And Suzanne is here, so I'll come back to that. Let me start by, on behalf of David and I can't say thank you enough to the Crafoord Foundation, Crafoord family and descendants, and to the Royal Swedish Academy for Sciences for this enormous honour. It really, tusen tack, and that's the limit of my Swedish, unfortunately. Thank you for celebrating what I think, and I think David thinks, is one of life's most beautiful and essential processes, B-cell tolerance. And I think that's really the thing that makes this special this morning, is it's a moment to reflect on the glory of biology and the difficulty that we as medical scientists have coming to grips with it. I also want to thank three brilliant mentors that really got me started in medical research and in this B-cell tolerance story. Now, the first of those is Gus Nossel at the Walter and Eliza Hall Institute. He was the director at the time. He was the original single-cell scientist and B-cell tolerance pioneer. He did his PhD with McFarlane Burnett, who I'll come to in a moment. The second is Mark Davis, who was at Stanford University as a young scientist, just set up his own lab, and really one of the vanguard of the new, at that time, field of molecular genetics and gene technology being brought to immunology. And he agreed to my request in a letter to work for free in his lab for 6 weeks to learn molecular biology before I started my PhD with Gus. I ended up staying for the better part of a year, and Mark even started paying me after a while. And then the third and most important is Tony Baston, who first gave me the opportunity to try my hand as a veterinary student in immunology research in his laboratory, and then to come back to his laboratory when it became clear that to make the transgenic mice, it needed to be in Sydney for reasons I'll explain, and it needed an interdisciplinary team which Tony had assembled in Sydney. And he gave me both the mentorship and the confidence to take on what really was a much too ambitious PhD project. But he never said no. And, now, the other point that I really want to make this morning is you may have heard of Planck's principle. Not Planck's constant, which has to do with wavelength of light, but the principle that science progresses one funeral at a time. My lived experience, come to grips with tolerance and autoimmunity, suggests the opposite, that science actually progresses one birth at a time, and it's the birth of new technologies. And I think this point is one of the reasons why, this morning and the Crafoord Prize is particularly special, because, as you've just heard from Olle, Holger Crafoord, was not just an entrepreneurial giant, but it was based on new technologies, whether it was Tetra Pak, which we all have in our houses as a way to have sterile holding of fluids, or a revolution in dialysis for people with autoimmune diseases like my mother had, who fortunately didn't progress to the stage of dialysis. So to have a prize based on brilliant technology that celebrates the use of technology to figure out how life works and make it better, it couldn't be better. So thank you. Well, what's B-cell tolerance? It really is how our body avoids making antibodies against ourselves. And as I said, it's really a process of dazzling beauty, but it took a very long time to see it in all its beauty. And you can see here a wonderful quote from a classic study from Polyarthritis, where through experiments that he did in goats and some experiments that he was aware of that were being done in Vienna by Landsteiner, that I'll come to, he deduced that there were these contrivances in the body that avoided making antibodies against ourself, autotoxins as he called them. And as you can see, oh, you can't see, sorry. And so in 1901, he said these contrivances, whatever they were, perhaps laws, some kind of physical law, were more important, were tremendously important, because the production of autotoxins, of autoantibodies, would be a danger threatening us, more threatening than any exogenous injury. But it did take a long, long time to see what those contrivances were. So they first started to come to view with Emil von Behring's huge advance of serum therapy, again, really before the 20th century. In serum therapy, this was the first time that medicine could target treatment against the causative agent, in this case, the strangling angel, diphtheria, which was a terrible scourge across Europe and the world at the time. What Behring developed was the way to give horse serum containing immunoglobulin, IgG, that neutralized the toxin of the diphtheria bacteria. And if it was given in the first couple of days, it could save the lives of children who would otherwise have a high chance of dying from diphtheria, suffocating to death. So it was a revolution in medicine. It was the beginning of targeted immune therapy, which has just gone from strength to strength in the subsequent more than a century. But it was a dangerous therapy. And the reason why is illustrated here, that when you inject horse serum into our body recognizes the foreign parts of the horse IgG, immunoglobulin G. It recognizes those parts of the IgG that are not identical to our own immunoglobulin. It recognizes them as foreign, and it makes antibodies against them. And those antibodies then trigger inflammation in the joints, in the blood vessels, in the kidneys, in the nerves, in the heart, in a disease that von Puke defined as serum sickness, in about 8% of people with horse serum. And for that reason, although we still use horse serum very rarely as a treatment for diphtheria, it's only as a last resort because of this problem of serum sickness and anaphylaxis. Now, by contrast, if you use for serum therapy human immunoglobulin, human serum and human IgG, it doesn't pose a risk of serum sickness. And this is where B-cell tolerance first really comes clearly into view. That in my state of Australia alone, from this paper, doctors use tons of human serum IgG, literally tons, are given to tens of thousands of people to prevent or treat hundreds of diseases. And the way our body accepts human IgG as self and doesn't make antibodies to it is the basis for a hugely important therapeutic. And it's also the basis for Australia's largest biotechnology pharmaceutical business, CSL, which is now a multinational corporation. And its main product is human IgG. Now, the B-cell tolerance to human IgG is also behind the explosion of new treatments for diverse diseases that are based on human monoclonal IgG antibodies. So as opposed to serum treatment with individual human or humanized antibodies that are produced by genetic engineering. And the first and greatest of those really was Humira, the antibody, one of the antibodies that Pan mentioned in her introduction against TNF as a treatment, hugely important treatment for rheumatoid arthritis and other autoimmune diseases. And the beauty is, the reason why Humira can be given over and over again on a monthly basis to someone with all these autoimmune diseases without triggering serum sickness is because of B-cell tolerance that stops us from making antibodies to our own IgG. But B-cell tolerance is imperfect. In some of us, often later in life, we start making antibodies against our own IgG. And these autoantibodies are called rheumatoid factors because they were first discovered in people with rheumatoid arthritis. In some people, they accumulate in sufficient quantities and with a particular quality that they trigger the same kind of blood vessel and joint and kidney and nerve and heart inflammation that looks very similar to serum sickness. And I'll come back to one of those diseases a little bit later briefly to understand at least one way that this failure of B-cell tolerance can come about. Now, B-cell tolerance also came clearly into view over a century ago with the experiments of Landsteiner and the discovery of blood types. And I think each of you probably know your blood type, whether you're A or B or O or AB. Knowing and being able to match a donor and a recipient is at the core of the success of blood transfusion. Again, something that medicine just can't do without. And the reason that works is because, and what Landsteiner discovered, was that we make antibodies against other people's blood types, but we normally don't make antibodies against our own blood types. So as long as you recognize that, you can safely transfuse. And the reason we don't make antibodies against our own blood types is this phenomenon of B-cell tolerance. But again, B-cell tolerance to our erythrocytes, to our red blood cells and our blood types is also imperfect. And some of us will make autoantibodies against our own red cells. And one of the first examples where this was really clearly demonstrated was for an autoantibody class that we call cold agglutinins. And it's worth just reading this brief description from this beautiful paper, which was the first clinical paper describing this disease, one of the first autoantibody-induced diseases. It comes from a man in his middle age in Minnesota, which I think is even colder in winter than Sweden. And he noticed that when it was really cold, his urine turned red because his red blood cells were being destroyed, as it turned out, as these researchers showed. When it was warm, everything was fine. The next winter, again, the urine would go red. His fingers and toes would turn purple. And sometimes he was a metal worker and he lost a couple of the tips of his fingers from frostbite, essentially. And when he ate ice cream, the tip of his tongue would go purple. And this was all because he was making antibodies against his own red cells that at low temperatures in the extremities were clumping the red cells together and then even either blocking the blood vessels or causing the red cells to burst and lyse. And this is not a common autoimmune disease, but still a devastating one even today. So what normally stops us, what normally keeps the peace? You know, we are constantly making antibodies, waging war essentially against all sorts of bacteria and viruses that are infecting us. And it's crucial throughout that antibody wars that we don't make antibodies against our own things. So how does it come about? Well, in 1945, Ray Owen, an American geneticist, discovered that twin cattle that are genetically dissimilar and have different blood types exchanged blood between one another during their fetal development through the placenta. And as a result, for the rest of their lives, even though they had different blood types, the two cattle, they didn't make antibodies against each other's foreign red cells. It was an incredibly important observation at the time. And so McFarlane Burnett and Frank Fenner, two Australian virologists, saw that and recognised what that meant. It meant that these laws that Ehrlich had proposed that existed to stop us making antibodies were not hardwired in our bodies. The immune system somehow learned during development not to make antibodies against self. At the time, it was not clear what that would be, and Burnett's idea of what it would be was at the time was different. But then a key set of experiments by British immunologists, led by Peter Medawar in 1953, experimentally demonstrated that fact, that if early in life the immune system is continuously exposed to foreign blood types, it learns not to reject them. Medawar coined the term actively acquired tolerance. It was a learnt, actively acquired process. And then in 1957, Burnett laid out the first theory of exactly how this learnt process to not make antibodies against itself might work. And so I'll walk through that very briefly. So Burnett's theory was beautiful for its simplicity, but daring in terms of how far it leapt forward in way of thinking, but most importantly, relative to any of the tools and technologies that immunologists had to test Burnett's ideas in 1957. So he proposed that during development, and later throughout, we now know throughout life, there was a mutator that mutated the antibody genes so that millions of different antibody combinations could be created. But the other key thing he proposed was that each different B cell would display one type of antibody only on its surface as surface immunoglobulin, membrane immunoglobulin as receptors, and that's denoted by the different colours here. And then what he proposed was that during fetal life or early B cell development, these antibodies on the B cells would be tested to see if they were harmful, if they reacted against our own blood types or our own IgG, for example. If they were deleted, purged from the repertoire forever. If not, they were kept, so that after birth, the only B cells you would have in your circulation would react with foreign antigens potentially, so they're potentially useful. And then his second part of his theory was that amongst those cells, if one of them recognized a virus or a bacteria that you were infected with, they would multiply that cell and make many copies of itself and then turn into an antibody factory that secreted those antibodies. And that would explain this ability to make antibodies against any infectious agent that are exquisitely specific for the virus, but not for self. Now, when David and I both independently came into this field in 1983, Burnett's theory had been discarded as false by pretty much the whole field. And that was summarized in this, in this beautiful review that Gus Nossel put together in the first edition of the Annual Reviews of Immunology, where he summarized that, much to his chagrin, having trained with Burnett, no one really believed Burnett's theory. And in fact, most people didn't believe B-cell tolerance existed. And there were various reasons for that. There was good evidence, including some very good evidence from here at the Karolinska Institute, from Göran Moller and his team, that self-reactive B-cells with self-reactive antibody were clearly present in healthy individuals. Gus Nossel had proposed a variant on it, clonal anergy, but it was clear that you could find cells that could be stimulated with mitogens so that really didn't seem to be possible. There were other theories that were very popular for a while until the tools of molecular genetics made them go away. And many of the people, as Gus summarized in 1983, said that, well, maybe the whole concept of tolerance is dead, and it left the field with no explanation, really, for all these striking findings of Ehrlich and Landsteiner, Owen, and Medawar. So, what to do? Well, the thing that really made the field stuck was that antibody mutator of Burnett's and the complexity of all of the different antibodies. It meant that Burnett proved to be right in that that mutator created millions of different antibodies, each B cell making a different antibody. But that meant that it was impossible to see what happened to one B cell in a million that recognized self-IgG or that recognized a self-blood type. You just couldn't see, you know, a needle in a haystack. And if the hypothesis was that that needle was deleted from the haystack, it was even more difficult. And so that really was the problem. And what came to reign, of course, was very much Planck's principle that, you know, the old guard held back the young guard, and whoever talked loudest for longest, they must be the right ones. So when I started my PhD in 1985, there were powerful new technologies that had been born to understand that antibody gene mutator mechanism. It involved, and to understand antibody diversity, tools to clone and sequence antibody genes, and most importantly, what had just emerged was tools to introduce antibody genes that encoded particular specificities back into the inherited DNA material of a mouse, a transgenic mouse. And that was very much, suddenly everyone was thinking along those lines, particularly for T cells, as was mentioned in the introduction. And I decided I'd focus on B cells, given that I was due to go back to Gus's lab from Mark's lab, and Gus was the world authority on B cell tolerance, and I could do an experiment to test whether he was wrong or right. And I always loved Karl Popper, the great scientific philosopher, who said that the greatest experiment you can do is one that falsifies a hypothesis. And so I thought, well, we'll see if we can falsify it. And so I focused on antibody genes which encoded what was then the best understood protein target of antibodies at the time, hen egg lysozyme from the egg white of chicken eggs. Very, very well studied, and by introducing the genes for antibodies against that hen egg lysozyme target, I could engineer mice where many of the B cells had the same immunoglobulin on their surface. So now we weren't looking for a needle in a haystack. Half the haystack or more was identical. And so if they were deleted or if something else happened to them, it should become clear and obvious what or nothing. If most people were right and there was no B-cell tolerance, it would be clear that that's the case. And I think David has a very parallel, independent story. We didn't know we were working on these two tracks at the same time, but same thing. So the idea was, as you heard in the introduction, was to cross these two strains. So introduce the hen egg lysozyme gene as a new mouse gene, so that lysozyme was now present in the serum of the mice alongside IgG. And so whatever explains tolerance to IgG, it we should see the same thing happen to tolerance to this normally foreign chicken protein in the double transgenic mice. Now, I guess the other key point to make, I mentioned technology is really at the core of it, and it was just a moment in time when that technology was finally ready to tackle this problem. But technology takes teamwork, and it was, I was really lucky to be a part of a wonderful team, and thanks very much to Tony Baston for making that possible. So let me just walk through that. Tony, of course, had an incredible training as a clinician and Australia's preeminent clinical immunologist and very strong experience in cellular immunology. I was very lucky to set up a collaboration with Sandy Smith-Gill and her PhD student Tom Lavoie at the NIH in Bethesda, Maryland. Sandy was the world authority on monoclonal antibodies to heneglysozyme and had cloned the genes for some of these antibodies and shared them with me in a collaboration. Now, the biggest challenge was that the technology to make transgenic mice was very new technology. It had only just really been established in a few labs in the United States, in particular one lab in New York City led by Frank Costantini and Elizabeth Lacey. And they actually ended up writing the textbook on how to do this. But at the time, there was no textbook. And I was in Melbourne at the Walter and Eliza Hall Institute, and this was absolutely on the critical path of this wild and crazy project. And I discovered that there was one person in Australia that had a lab that was making transgenic mice. She'd just returned from a postdoc with Costantini and Lacey in New York, and she was already making them. But she was up in Sydney, in the western suburbs of Sydney, and that was Kathy Raphael. And so I went up and visited her and said, if I moved back to Sydney, could I come out and learn how to make transgenic mice? And very grateful that she said yes. Now, I was much too impatient as a micro-injector down the microscope to inject DNA into mouse eggs. I would burst more mouse eggs than anything. But one of the great things Tony did was pair me up with a really gifted technical scientist in the lab, Jeff Crosby, who was really good at injecting mouse eggs. I taught him how to do it, and he immediately was 10 times better. And had that not happened, we would have been injecting for the next 10 years to get one mouse. Now, once you have these experiments, you need all the old tools of cellular immunology. And Helen Pritchard-Briscoe in the team, she knew how to do Niels Jörner's plaque assay, one of the foundations of measuring antibody-forming cells from the 1950s, and that was crucial for the experiments, and how to do adoptive transfers and make bone marrow chimeras. All this sort of voodoo of cellular immunology was all in place. And then the most important readout to see what happened to these B cells was, again, a rapidly developing new technology of flow cytometry. At this stage, Tony Baston had gotten a big grant from the government in Australia to get the state-of-the-art flow cytometry machine that could do measure 3 different colours on one cell at the time. Great big 15-inch floppy disks to, you tell students now what was the cutting-edge technology, and they have a good chuckle about it. So the point being, it takes a team, and I was really lucky to have a team, and it takes lots of different technologies. So what did we see? Well, what we saw with the new technology was a real surprise, and we saw 2 things. First, B-cell tolerance was as clear as day. When we measured the serum in the mice, the B cells had stopped secreting the antibody in the double transgenic mice where lysozyme was self, and that's a log scale there. So there was a 50-fold reduction in the activity of the B cells in terms of secreting antibody into the serum. So that was great. That told us B cell tolerance seemed to exist, at least in this very artificial experiment that we were doing. But the second thing was that when we sat down on that flow cytometer late one night, what we discovered was that the B cells, although they were tolerant, they had not been deleted. But they had changed in a curious way that no one had ever anticipated. They downregulated one of the two flavors of immunoglobulins that the B cells display on their surface to test whether these receptors are harmful or useful, IgM, but they'd kept the other one, IgD, and so you can see that in the double transgenic mice there. Now, this was a pretty weird outcome, and the first thinking would be that maybe this is all an artifact of this very artificial experimental system that we'd engineered. But the one thing that argued that it might not be an artifact is that normal B cells in mice and in humans, most of them actually look more like the tolerant B cells than like the ones that weren't tolerant. And so that led us to propose that, and we were rapidly able to do experiments to show that there was a very tight relationship or correlation between downregulating IgM and stopping the antibody secretion. So we proposed that this might mean that a large fraction of B cells circulating in normal mice, where you hadn't done all this genetic engineering, were also doing this process of tolerance. So I presented this, I didn't yet have my PhD, but I presented it at one of the world's biggest conferences in Cold Spring Harbor in New York. And the first question came from the great Av Mitchison, who said, I don't know why you've done these experiments when everyone knows there's no B cell tolerance. And Av was brilliant. I'd read his papers and I knew where he was coming from. But there you go. That is what we were up against. And so in the ensuing several decades, we had a lot of work to do. And I was lucky that David also found B cell tolerance existed. There was tension because David's mechanism was different from ours, and that fed people like Av Mitchison who said, well, the whole thing's just a transgenic artifact and will go away. And but over the ensuing decades, the birth of many new technologies has enabled David's team and my own team and many others to decipher the negative signals that are actively stored by the B cells, depending on the degree to which their surface immunoglobulin is self-reactive. And that's not a simple all or none thing the way Burnett was thinking. It's shades of grey. And what came to pass was that if the cells are moderately self-reactive, they store a negative signal, but they're kept. If they're strongly self-reactive, as David will elaborate because it's really his work, then they arrest and do more mutations. But these other ones are kept, even though they're potentially harmful because they might be useful. But they don't secrete their autoantibody. So the other problem that I faced at that Cold Spring Harbor meeting, the next question was this one. It doesn't make sense, Chris. Why would the immune system want to fill itself with B cells that can't secrete any antibody, that are anergic and unresponsive to any stimulus? Now, our experiments pretty much straight away indicated these cells weren't anergic in the way that Gus Nossel had defined, where they couldn't respond to an antigen or a mitogen. They proliferated just fine to LPS, the cell wall component of bacteria, but they just didn't secrete the antibody. And even more curiously, they proliferated even better to a cocktail of anti-CD40 and interleukin-4 that is the signals that a helper T cell provides a B cell. These tolerant B cells actually proliferated better than their non-tolerant ones. So they weren't tolerant. So when we made these observations, and others like them, we did propose that maybe the reason the system was keeping them was that the cells could mutate their antibodies if they reacted to a foreign virus and select progeny cells that had mutated to lose self-reactivity and were now virus-specific. Now, I have to admit that we put that in a couple of papers as theoretical things, but I didn't take it seriously. So again, this is Planck's principle, but now the shoe is on the other foot. I'm suppressing it, but mainly because we just didn't have the tools. Those transgenic mice were not up to the task. And it really wasn't until 2014 with second-generation transgenic mice that we could come back to this problem and approach it seriously. And we did that initially with the lysozyme system. I don't have time to walk through that, but I would just like to take you briefly through an unpublished summary of some work that I think ties this all together, for me, very beautifully, and brings us back to cold agglutinins. And so this is with third-generation immunoglobulin transgenic mice, that actually the story starts with the vaccine that is the greatest vaccine of all time, the one that eradicated smallpox, vaccinia virus, and a healthy volunteer who had received that smallpox vaccination 30 years prior and then was boosted, and blood collected 14 days after his booster, and antibody genes with the new technology at the time were cloned from individual cells in his blood. And one of those antibodies is the one that we've studied in this particular series of experiments called 589. And 589 recognizes the virus, and it doesn't recognize self. But when we reverted the 27 mutations in the antibody gene that were present in that single plasma cell, the precursor antibody, pre-589, recognizes the virus more weakly, but it also is representative of one of those cold agglutinins that I mentioned before, those original autoantibodies. So it binds the erythrocyte I blood type, a self-blood type, and agglutinates them potentially, but it also binds an integral virus protein that's completely different. So it's an antibody that binds two very different things. So then, using the new technology of CRISPR-Cas9, we were able to make, and we meaning Rob Brink, was able to make at Garvan a third-generation mouse targeting these genes into the correct position, and then to see, well, what happens to B cells that express a human immunoglobulin that's representative of 6% of circulating human B cells that has the potential to cause a cold agglutinin if it was secreted. And without going through all the details, let me just summarize what we found, which, to me is maybe the most beautiful result so far, and we're just writing it up. So in these third-generation mice, the mutator system is still in place, but we've hot-wired it, if you like, so that many of the B cells make pre-589, this antibody that has this harmful properties of self-reacting and good properties of virus reacting. And those cells are not deleted, but they come out and store negative signals very much like we see with Hennig lysozyme, although now that's due to recognizing the self-blood type. And they don't secrete the autoantibody. And then we gave the mice the vaccinia virus to replicate what had happened in the original human donor with the smallpox vaccination. And what we find is that now we're essentially testing the B cells for their ability to, the antibody to bind vaccinia virus. And what we find is the B cells are not anergic and unresponsive. They proliferate and mutate like crazy, mutating their antibody genes in germinal centers. And within 2 to 3 weeks, all of the progeny of these cells have found mutations that get rid of the binding to the self-blood type, but then improve the binding to the virus. So it really is, to me, amazing that biology and our bodies have come up with a system that in a space of a couple of weeks can evolve an antibody from a bad antibody to an incredibly good, exquisite, specific antibody, and that's, of course, the magic of antibodies. Now, let me just come back in the last slide or two to this question of rheumatoid factors and the breakdown of tolerance in that leads us to make antibodies against our own IgG. Now, one of the groups of people where this happens most frequently are people with a chronic hepatitis C virus infection. So 50% of people with chronic hepatitis C virus will make antibodies against their own IgG, rheumatoid factors. Now, in most of those people, that's something you see in the laboratory, but doesn't actually cause symptoms of serum sickness or rheumatoid arthritis, etc. But in 10%, it actually creates of vascular inflammation of the kidneys, that damage the kidneys or the nerves, what we call cryoglobulinic vasculitis. And so this is one example of one of these diseases, but where we know what the original trigger is, a virus infection. And so, to cut a long story short, in a paper we've just published, we've been able to use the very latest new tools that I could only dreamt of 10 years ago, of single-cell sequencing of blood cells from patients with this disease, to piece together why this disease develops so frequently and why B-cell tolerance breaks down so aggressively in people with this disease. And essentially, it's all about mutations and this mutation game that the B cells are playing. So the first thing is VDJ recombination, the process that initially generates antibody diversity. About 5% of the B cells that arise from that have antibodies on the surface that bind our own IgG. But what we find is often with very, very low affinity, below the threshold where these B cell tolerance mechanisms kick in. Then, if you've got hepatitis C virus, millions of virus particles are in your bloodstream coated with your own IgG. And so it's a self-protein, but decorating a very immunogenic particle that confuses these B cells into activation and proliferation. And they start to mutate, but because they're now responding to self-IgG on an immunogenic particle, there's no way of mutating to lose their self-reactivity. And so they, in fact, increase their affinity to self. But also they acquire thousands of mutations, not just in the antibody genes, but across all the chromosomes. Because that's the downside of this mutation mechanism, is it's not as perfectly targeted as you'd like. And so what happens in 50% of people is just that alone is enough to trigger a laboratory-measured autoantibody, a rheumatoid factor, but it's subclinical. But in the people that develop the full-blown disease, one of those thousands of mutations elsewhere in the genome provides one cell a growth advantage or a survival advantage, or what we call a lymphoma driver gene mutation. Not enough to become a lymphoma or a leukemia, but just enough to give that clone an advantage, and it makes more progeny and more plasma cells and more autoantibody, and now the levels of autoantibody reach a threshold where they precipitate vascular inflammation and essentially disease. So really then, just to wrap up, B-cell tolerance turned out to be much more complicated than Burnett's original theory of clonal deletion. Whereas the simplicity of Burnett was, if in doubt, throw it out. Delete those cells if they have any risk of being harmful. What I think we've learned is that it actually works by waste not, want not. That you really, our immune system uses mutations to turn bad antibodies that could be harmful against our own IgG, against our own blood types, into good antibodies that are exquisitely specific against a virus or a bacteria. But the way it does that is to play a mutation game, and that's playing with fire. So it plays this mutation game early in life. Once we're infected, it plays the mutation game again, as I've described in the germinal center. More mutations. When that's working perfectly, and for most of our life, and most of us it does, everything's fine. We only make antibodies against a foreign virus. But in some of us, at some point or another, for a variety of many different reasons, one or more of our self-antigens become immunogenic in the way that I described for the hepatitis C virus as one example. And then some of these mutations take that to a point where that precipitates autoimmune disease. And what I'm very excited about at the symposium in Stockholm, we will hear some more developments of new ways to essentially clear out all these mutated B cells and cure potentially autoimmune disease by essentially rebooting the system, getting rid of using antibodies or CAR T cells, removing all of the mutated B cells that have the wrong antibodies because of mutations, etc., and then allowing a new crop of B cells to develop, which may reset the immune system to such a degree that what we're starting to see, for example, from some of the experiments at George Schett in Germany, patients with lupus where nothing has worked, that are now completely off all of their medication, even prednisone, which is something which 3 years ago I would not have even dreamt was possible. So I think it's very exciting times, and that's another reason why I'm so thrilled that the Swedish Academy has made the decision to celebrate this process today, because it is a beautiful process. It's taken a long time to see, but I think now we are seeing extraordinary fruits from understanding and harnessing the process. Now, I can't even list all the people that have worked with me in the experiments that I've summarized today. These are just the people in these unpublished or recently published studies that are summarized. But I guess the other important person to thank, most importantly, is my wife, Dr. Suzanne Hartley. You couldn't have a more wonderful partner in science and in family and in life. So, Suzanne, thank you. And I'll stop there. Thank you.